Thank you. Although I'm not a doctor, I'm a student, but that's okay. Good morning. I come from the University of Oxford, and I'm going to speak about the GWAS in UK Biobank. I will start with a bit of a background on the UK Biobank, which is a cohort study with 500,000 volunteers, aged 40 to 69 years old from the whole United Kingdom. And we have gathered questionnaires and biological samples and also physical examinations from all these volunteers. And this resource is also connected to the long-term health records from the National Health System in the UK. It's important to note that all the participants are characterized prior to having any health outcome. And this is a prospective epidemiological study, so it's not focused on just one disease. We got an imaging extension, so we are bringing back 100,000 from the volunteers, and we are acquiring images, cardiac images, abdomen images, and bone density, among others. And the brain images modalities that we have are 6 different modalities. And so far, we have scanned a quarter of the total, 25,000. About the genetic information. We have genetic samples from the whole data set. And the original array of data is 820,000 SNPs, and these have been imputed to 96 million genetic variants. And all this data was released last July. Well, July is last year, sorry. I'm going to talk now about the brain imaging part. We have 6 different modalities. The structural modalities are T1 weighted, susceptibility weighted images, and T2 FLAIR. For diffusion, we have 100 different directions in 2 cells. And the functional images are resting state and task fMRI. And what are we doing with these images? Well, starting from the raw the raw data, these images go through an automated processing pipeline, based on mostly on FSL. And out of that pipeline, we get a set of processed images in nifty format and what we call IDPs, imaging-derived phenotypes, which are summary measures that describe a feature of the brain. And I'm going to give one example. The volume of white matter hyperintensities that we get from the FLAIR images using a tool developed in our lab called Bianca. So what can we do with an IDP like this? Basically correlated with other measures, like, for example, this is the known relationship between the volume of the white matter hyperintensities and the age. But this is just one association that we can find. And actually, we have run 40 million univariate association between IDPs and non-brain imaging variables on the first batch of 15,000 subjects that we have processed. In the x-axis, you can see the different non-brain imaging variables, in the different types of non-brain imaging variables, and the y-axis is the p-value of the relationship, with some values being higher than 10 to minus 100. But as you can imagine, with these numbers, it's very important to deal correctly with the confounds. And the first idea that is important to have in mind is that one man's signal is another man's noise. And what does it mean? That, for example, for some biophysical measures, some of them may be affecting and be confounds for IDPs, but also be very interesting for some studies. One example would be the blood pressure or the heart rate, which directly affects the IDPs, but is also related with disease and with aging. And the same happens with the head motion. With the head motion, as you can see in that image, everybody knows that motion can affect the images, and it's a strong confound, but it's also related with some diseases and aging. A different type of confound are the changes in the scanner or the drift in the coils, the quality of the coils. And one example of these kind of changes is the head position in the scanner, which is an issue that has been taken into account with dealing with the confounds. And for the genetic information, the way to deal with the confounds is very standard in genetic studies, which is removing the 40 principal components from the data. And now I'm going to talk about, if you want to know more about how we are dealing with the confounds in brain imaging, you can see poster 2368, I will be there. Now I'm going to talk about what we have done with the genetic information and in relationship with the brain images. We started with 8,400 subjects that had a common genetic ancestry. And from the initial 96 million genetic variants, we have reduced it to 11.7 million SNPs, by applying different filters, like only taking the variants that had a p-value for the Hardy-Weinberg equilibrium higher than 10 to minus 7, a minor allele frequency superior, higher than 1%, or the insertion information score higher than 0.3. And we are just keeping the SNPs variants that were included in the list by the haplotype reference consortium panel, which is like the authority in this kind of studies. And we have run 3,144 different GWAS, one per IDP. And what we have found is we have found 148 significantly associated clusters, and we have replicated those clusters in a different set of 3,000 subjects. This is a Manhattan plot that is the typical result of a GWAS study. And once we identify the SNPs that are significantly associated with the IDP, we can run a GLM analysis with that SNP, and we can get what parts of the brain are different in the two groups, like having a certain value for a SNP or another. And all this information that we have found is available in this server, the Brain Imaging Genetics interactive server, for anybody to check. Another interesting fact that we have found is the 50% of the IDPs have a significant SNP heritability, especially in the structural IDPs. The cortical volumes gotten by using FreeSurfer have especially high heritability, but also white matter hyperintensities. For diffusion IDPs, the heritability is in general high, around 50%. And for functional IDPs, the heritability is not that high. But after we summarized the functional connectivity IDPs, the heritability was higher. And the way that we summarized this was, we have almost 1,700 different functional connectivity IDPs that basically those are the edges from a network connectivity study. And each edge is distinct, but it's relatively noisy. So the heritability is very low. That's the reason. So the way that we solved this was by joining all these IDPs together in a matrix with the subjects and the IDPs, and running an independent component analysis. We got at the end 6 different connectivity features, and those features result in a much higher heritability. And as you can see in this box plot which I saw before. But these summary features are also have a biological meaning. Here we are showing 3 of the 6 features, and the 6 strongest, the 6 edges that are more strongly involved with each feature. And for example, for the first independent component, all these edges belong to the sensory motor area of the brain, For the second feature, the 6 more strongly involved edges belong to the cognitive areas, and for the last feature, into visual areas, and that's an interesting result. Now I'm going to show you 2 examples of some of the clusters that we have found that are significantly associated with some IDPs. In this case, the IDP is the mean intensity of the T2 star image in the putamen. Sorry. And we found that there was a SNP in the 3rd chromosome that had a p-value of 10 to minus 22. But in the same GWAS, we saw that there were another 3 SNPs that were very highly correlated with that IDP. And those are what we call the cluster of IDPs. Of SNPs, and those are the 4 SNPs that are highly correlated. And so we ran a GLM analysis with these, 4 GLM analysis with these separate SNPs, finding that each SNP affects a part of the putamen in a different way, and that is a really cool result. It is known in the literature that the T2 star shows how the iron is deposited and how that is affected by aging and by diseases. And it's also known in the literature how the genes that these SNPs belong to are associated with iron transport deposition. So we are showing what is the relationship between imaging, diseases, and genetics. And finally, this is another example of another of the GWAS that we ran. The IDP is the gray matter volume in the hippocampus obtained with a VBM analysis. And there was a SNP that was very highly significant in the 12th chromosome. The p-value of this relationship was 10 to minus 13.2. And this SNP is associated with the gene HRK, which is known to be related with is known to be expressed in the hippocampus and also related with apoptosis, the cell death, and with Alzheimer's disease. Once we ran the GLM analysis with this SNP, we can see that the differences in the group are strongly in the hippocampus. As you can see, this would be an interesting biomarker for disease. So the take-home message would be that the biobank can be used to find interesting relationship between these 3 elements: diseases, genetics, and brain imaging. But that's just using this part of the biobank, because the biobank is very rich and you can find more relationship with many other sources of information. And that's it. Thank you.